very willing. So today we're talking about dynamics. So we sort of already talked about some type of dynamics, right? So pseudo time is one way of looking at things. This picture we gave last time, right? We assign every cell some sort of pseudo time. And so today we're not doing pseudo time. We're doing something slightly different. And I'll explain what that is. So here's one way of visualizing something. I have some number of cell states over time, and here's my picture of cell state, and this is how we often look at it. Maybe we can look at it broken out by time point. That may be useful. And so, right, if I tried to pseudo time on this kind of thing, then maybe I would say, the bottom is the first pseudo time and the last is the top. But that's not really what's actually happening here, right? So if I assigned, if I took the middle pseudo time, then this is not really anything to do with actual time. And so today we're sort of modeling actual time instead of pseudo time. So the goal is to predict middle time points or interpolate maybe, right? So, one of the questions you could ask is, right, like given a cell at some time, what is its location in transcriptome space in a previous time or a future time? Another question you could ask is, given, right, sometimes what does the cells look like at the time in the middle? And so, right, pseudo time sort of gets at this, but it's slightly different, right? So pseudo time gives you the discrete days as your data, and then it assigns days, pseudo times per cell. And so, you could say, right, at 0.25, we have sort of this cell distribution, but that's not actually what's happening, right? So, you may have too low variance, right? Because what we'll really have between day 1 and day 2, right, is some sort of blending of day 1 and day 2, right? The most advanced cells maybe continue on up and these continue over. And so we'll have sort of a spread somewhere in between these 2 days. And so, we're modeling the actual time instead of the pseudo time here. And so we're going to go into differences between probability distributions before we talk about anything else. So one sort of difference that you may have heard of is the KL divergence. And so the way this works is it's basically a sum over bins, you can think about it, or sum over, up and down axes, right? So you take the amount of things in P and you divide by Q, and you take the log and you multiply it by P. And so what's important here, right, is that you're sort of comparing bin-wise up and down. And when 2 things are exactly the same, then the KL divergence is 0. And when they are very different, then it goes to infinity. And so this, right, is somewhere in between the 2, right? Lots of bins overlap, some are slightly different, but as these Gaussians get closer and closer, the KL will go to 0. But let's consider a different case. So what if we have 2 Gaussians and they're very steep? And in fact, their support basically doesn't overlap. So what is the KL divergence between these 2 distributions? Well, we look at the bins and we look at, P divided by Q. And we look, say here, right, P divided by 0Q is going to be a very bad number, very large number. And so we will get essentially that the KL divergence here really goes to infinity as these things get separate support. Am I missing? No, okay. Excellent. And so we can take this to the extreme, right? So, okay, we have 2 distributions that are extremely sharp Gaussians that are very, very close. Well, still the KL divergence is infinity. And so, you might think, well, these distributions are quite close together. And so the thing we'll be talking about today is optimal transport. And it's a different way of measuring differences between probability distributions that says that these things are actually quite close. And so, right, vertically, these are vertical distances. We're going to be talking about horizontal distances. It's one way to think about it. And so optimal transport, one way of characterizing it is also called the earth mover's distance, because it's sort of like moving a pile of dirt from one place to another. So if I have if there's a distance or a cost between any 2 points, then my total, my sort of optimal transport distance is how much mass I have to move to get from one distribution to the other. And so this is just the fancy math way of saying it, right? So if I have a distance, here's the Euclidean distance. I basically integrate over the joint distribution between the two. So, right, how do I move mass from here to here? That's just how you can think about it. Yeah, and so this is a very horizontal thing, right? So as these distributions get closer and closer together, the optimal transport distance goes down and lower. And so it doesn't have this problem where if you have two very steep Gaussians next to each other, their divergence is very low in the Wasserstein space, but very high in the KL space. So yeah, this is sort of a nice property. And just one of the problems with Wasserstein distances is actually very hard to compute. So, there's many works trying to figure out how to compute this thing, whereas KL divergence is very easy to compute.
Just to put that in a little bit of perspective, preparing one of the notebooks for the lecture, I think on like Wednesday of last week, I messaged Alex in the evening saying, I have two distributions over a single cell data set. I'd like to calculate the optimal transport distance between the two. The code was relatively straightforward. It just took like 16 hours to run. And that was just over like 2 samples from 30,000 cells. KL divergence between those two would take about half a second to calculate.
Yeah, and that's because, right, so KL divergence, basically you bin the space and then you just compare vertical distances. So, binning the space is easy and comparing vertical distances. So sort of talk about exactly how, what this means in the next slide a little more carefully. And so one of the ways you can think about this is your distribution is on the rows and your target distribution is on the columns or something like this. So, I have a distribution P at time 1, and I'm trying to get to a distribution at time 2. And there's a matrix that is the costs, maybe, right? So the cost between going from this point to that point, right? What is the cost from going to this point to that point? And then this pi is the joint distribution. And so it sort of says in this cell, how much mass is going from xn to y2, something like that, right? And so, if you looked at the column, right, you're looking at the mass at y2, where did it come from, right? So maybe it came from a lot from xn, maybe it came from a little from x1, maybe none from the others, right? So, and this is why it takes so long, right? So, Dan is talking about 30,000 cells, right? This would be a 30,000 cell by 30,000 cell matrix, and you're trying to, find a cost over that, right? 30,000 is just large numbers, so scales of n squared, and you have problems as you get large. And so, right, one of the things you can think about this as the couplings, right? So, where does this cell go in the next time point, right? So this sort of, over time, right, we could look at the pairwise optimal transport. So, this cell goes to here, then it goes to here, then it goes to there. And this is one way of looking at it. And so the paper that originally did this, they titled it Waddington OT. It was published in Cell. I think we briefly touched on this, but right, this is very useful. And what you do, right, is you calculate the Euclidean distance between cells, and then you calculate the couplings, and you can use these couplings to look at the ancestors and the descendants of any cell in a time series, right? And so this is really modeling over time. So what you can also do, right, is this, since you get a coupling, you can interpolate anything you want. So between time 1 and time 2, you could get a time 1.5 by sampling from time 1 and then following the paths and taking sort of a midpoint here of the paths. But yeah, so I'm going to talk about a thing that is doing a similar sort of distance, and it's very related, but it's doing it with neural networks instead. So if you remember the autoencoders that we talked about today, so right, it takes an input, does some sort of bottlenecking, and then gives an output and has the reconstruction error. So this is very similar to the models we're going to use, except there's a small difference. So what we're going to be using is invertible neural networks. And so what that means is that we're going from input to output the same way, right? We're doing reconstruction error. However, what we do instead is have all the layers be invertible. So neural network layers are not normally invertible, right? So if you have a matrix that is, if you're going from 10 dimensions to 5, that will never be invertible. You have to go from 10 dimensions to 10 dimensions. And so, things, right, invertible transformations, you can think of, right, rotations, flips, scalings, right? So some of these things, right? But general neural network layers are not invertible, and so you have to use special layers to use these invertible neural networks. But yeah, these are very cool, right? Because you can go from the output back to the input. So there's one output for every input, there's one input for every output. And so this is sort of the basis of normalizing flows. So one of the big problems in neural networks is modeling probability distributions. So if I have a very complicated time one, again, my complicated thing is not very complicated here, but you can imagine it being very complicated, right? So the distribution of images or something, then I can take a very simple probability distribution and I can apply a normalizing flow and try and match it to the more complex one. So, and so the key here, right, is that I can, for every input, I can find the output. For every output, there is one input. So I can, for an output, find exactly where it is in the sort of the time zero or the, the simply defined distribution, and see, right, what the probability is there. So if I map from here to here, right, it's sort of like, okay, here's the probability sampling it, and here's the other one. And so there's a little problem with this, which is the change of variables. And so I'll talk about that just a little bit. And so, right, so change of variables, this is the calculus thing, but, say I'm mapping a probability distribution that is uniform on 0, 1 to one that is uniform on 1, 3, right? Well, these are probability distributions, so the area sums to 1 here, right? And so if you looked, right, these are invertible arrows. And so you could find this location, but the probability isn't the same, right? It's sort of, it's half, exactly half, right? And the reason that is because sort of, like if you look infinitesimally, right, this arrow maps the 2 places over here, right? Because you're sort of expanding the space. And so that's sort of what the change of variables is about. So as long as you can do this, right, now we can find exactly what the probability is here just by inverting it, finding the probability and finding the change of variables. So that's sort of how the normalizing flow thing works. So, you can make this as complicated as you want, right? So for example, what people often do is just keep stacking these things, right? So as long as you have an invertible thing, you can just, right, make another invertible thing and stack it on top of each other, right? So, maybe you have one transformation, and now we can just look at, n transformations together, and, the change of variables rule just propagates. So you can keep summing along the log determinants or multiplying everything together to get the change of variables from the beginning to the end for any number of layers or networks or however you want to think about it. And so the thing we're doing here is going to what's called continuous normalizing flows. So we take the deep normalizing flows and just take it to the extreme. So what happens if I have, well, an infinite number of little transformations, right? Very small transformations. Well, this is, we can express this sum, this infinite sum, as just an integral over time. And now we parameterize slightly differently, right? So we have this function f that is, a function of the input and the time, and it gives a small dx, right? So it tells you, exactly at this time for this number, where are we going, right? And so we can do exactly the same thing and integrate the change of variables. It all works out very nicely in the math world. But this is sort of confusing. So what does this actually look like, right? So now we have this continuous distribution and a more complicated one, and we're looking at, right, slices. So looking here, we sort of have a vector field, right, at every time. And so when we integrate from time 0 to time 1, where do these vector fields end up? So it's a vector field that's changing over time, but you can map from one distribution to another more complicated one. And so this is the basic model for trajectory nets, mapping from simple distributions to more complicated, or mapping over time continuously. Yeah, so another way of thinking about this neural network, right? This neural network F, it maps from a cell state and some time to something that is the same dimension as the cell state, which tells you sort of the instantaneous change in that cell state, right? So thinking about maybe RNA velocity, right? RNA velocity also tells you, where is this cell headed? Well, this neural network also tells you where is this cell headed. But the key difference here, right, is that it takes any cell, even if you didn't sample it, right? So anything in the gene space, it'll tell you, given a time, where is that cell headed next? And so the goal here is to train this model to map over time the correct distributions. But the way, right, so you take a very small time step, you get a change, you add it to X, and then you feed it back in. And you do this over and over. And so that in that way, you can sort of integrate this thing over time as a normalizing flow and find, how does your X flow over time? So another way of visualizing this, right? Okay, we take a very simple distribution, a Gaussian in 2 dimensions, and we want to make a more complicated one, something that looks like a maple leaf. And so here's a video of what happens, right? So we have this density field, and it slowly changes over time according to this vector field that tells you where things go. So right, so this is saying at this specific time, where's the cell head? Where is this little mass headed? It is headed in this direction, right? And so the goal is to train a network that sort of is the vector field, right? The vector field takes a position and a time and tells you the sort of instantaneous change. And so yeah, that's the goal, right? But training this thing is kind of interesting, and so that was the main project here. So yeah, but here's the thing, right? Continuous normalizing flows, we have no constraint on sort of the paths you take. So, right, so you train these things and we make them match the distributions, but they can flow in any direction they like as long as you go from one time to another. And so, you can get all these weird squiggly paths. You may or may not get them. It depends on what exactly you give them. But that's not what we want, right? So one assumption you can make is cells flow sort of kind of efficiently over gene space, right? So, there's some wasted effort, but you don't randomly create transcripts and delete transcripts for no reason, right? It's sort of efficient as possible given other constraints. So we're modeling things as straight lines, basically. So, right, you want sort of to map the cells from one to another, but you want the lines to be as straight as possible. And so the way that you can think about this is you want the shortest set of paths possible, right? So that's sort of, that's awesome thinking, right? Bringing it back to optimal transport, optimal transport was looking at the set of paths between 2 distributions and trying to minimize the total cost. Well, it's doing the same thing, right? Trying to minimize the total cost of paths between the 2. And we can make this very explicit if we want, right? So I don't know if anyone cares to understand exactly this part, but there's a link between the Wasserstein distance and the dynamic sense. So there's this theorem that says, right, the Wasserstein distance, you can think about it, this is just saying you can think about it as the set of paths going from one distribution to the other. This maybe is the easiest way, right? So, okay, so I sample an X from my previous distribution and I integrate it over time. And this is saying I want the cost that is minimum, right? So I'm looking at the path. This is saying the average cost of the path going from here to here should be minimized. And so you can look at this slightly differently, right? So this is looking at it from a particle that is moving over time's perspective. And this is looking at it for a little point in space time. How is the probability changing? These are equivalent definitions. And so the main thing, right, is 2 things. I want the time 0 mass to be mu and the time 1 mass to be nu. And I can't create or destroy mass. So, if you follow these rules, then you must, and this optimization, then you will get the exact cost that is the Wasserstein distance, which is the Earth mover's distance. So it's all very equivalent in some sense. And so when you look at the continuous normalizing flows, you actually get an approximate dynamic optimal transport. The only thing we do is replacing this constraint, this hard constraint that the end probability distribution is exactly nu, with the softer one that says the KL divergence between our predicted distribution and the true distribution is as small as possible. And so, this is the optimization, right? This is the loss function that we use in the neural network. And this gives you a flow that is sort of optimal, right? Sort of optimal in the sense that it's taking the shortest paths. so right, just going back to this example, some of these, if you looked at these particles, right, we're looking for sort of the minimum cost. So we want all these particles to end at the sort of the place that they're going and go there directly. And this doesn't happen in general. It just, right, this sort of looks like it is doing it, but it's not actually. And so here's sort of a more illustrative example of what it might look like. So let's pretend we're taking this Gaussian and now transforming it to this S shape. Well, the optimal transport, right, is for, these are sort of showing the paths, right, from red to blue, is to take each one of these points and transport it to the closest point in the S so that they're all, so that you match the. But when you take a continuous and one-length flow, the paths that take are not these straight paths. They're sort of taking these roundabout paths. And so, if you add this regularization that we call the energy regularization, then you get these straight paths again. And so, yeah, so now we have a neural network that can do the same optimization as dynamic optimal transport, which is extremely hard to compute, right? So, in Dan's case, it was, right, 30,000 cells. It's no longer feasible. But neural networks, right, are very special in that you can take many batches and you don't need to look at all 30,000, right, 30,000 by 30,000 distances. So this can be much more efficient especially for large data sets. And so, right, we've seen this data set before, which is the M-bird bodies, right, 5 time points. And so what you might get there is this density. So this density, right, what does the density look like at any time? Well, we can compute it with this neural network by integrating a certain amount. So this is just taking cells at time 0, integrate them a little bit, over and over and over again, and you can see the density that it computes. So this is very interesting. Yeah, so, but right, we've sort of somewhat constrained things to be as minimum energy, but we also know a lot of other things about cells. So, right, they divide and they die. They can only sort of travel along these low dimensional spaces. So they don't travel in Euclidean space, which was what was assumed previously. And since they're destroyed, we can't measure them right at every time point, but we can look at the transitions based on RNA velocity, right? So RNA velocity says this cell moves a little bit in this direction at this specific time. We can integrate that into our model. So just some ways that we integrated things. So unbalanced transport is you allow mass to grow and shrink at every point. So in the standard optimal transport, right, if there's more points up here and fewer points down here, then you've sort of got to find a pairing that says the mass has to be conserved. So you get these sort of long paths that go between the two, but pretend these were cell clusters. You have maybe you could think about this as a growing population and a shrinking population, at least in relative proportions. Well, this is not, right? This population may not turn into this population. You may have the case where these cells are growing and these cells are relatively dying, or something like this. So we can actually incorporate this into our model very easily because it's a neural network. We can just add a regularization and another network that predicts where things are multiplying and dividing. The other thing, right, is that in this data we find the trajectories, and really, right, cells don't go straight from here to here. They follow this long curve. And so, now we're in neural network land, so we can enforce everything with regularization. So we could turn this into, right, this path, along my path, I should be close to other points. This is what this says. So just, right, I want to stay next to other points, which means that all the paths follow the density of things. And finally, right, velocity, well, we have sort of an arrow at every point. So now we can, match, right? We have a model that says what is the derivative every time point. Well, here's sort of more knowledge, right, on the derivative at certain time points, right? So we know for this cell, what is its derivative at this time? We can use that in the model too. Yeah, and so just a toy example. What's happening here, right, is that I have a distribution that's moving over time on a sort of a one-dimensional manifold in 2D. So, cells line this low-dimensional manifold, maybe over time it moves. And so the goal here, right, is I have cells at time 0 and cells at time 1, and maybe information about the velocity, and I want to impute the cells at time 1/2. So I don't give the model these cells, and I try and figure out, right, I want to match this distribution, and that distribution. What does it look like at time 1/2? So if you just do a regular normalizing flow with energy, right, the dynamic optimal transport is the same thing. It sort of just moves these and moves them into 2 separate piles because the orange is 2 separate piles. But when you say, well, I want to remain next to other points, it sort of bridges the gap. And when you know the velocities, which is even more information, you can have it follow this arch very nicely. And so, what we showed, right, is that you can, with these additional things, you can follow the actual dynamics of cells more closely. And so once you have these dynamics, right, you have for every cell, what is its ancestry and what are its descendants. So you can look, right, we have maybe some endpoints. We can look at the genes over time of that specific cell, which is something you can't do, right, if you have just 5 distributions. So this is, I think, very powerful. You can figure out, right, the purple cells were high in this late and the green ones are low, right? And so this is one of the big uses, right? Once you model the dynamics instead of the pseudo time, you have a picture of all of the cells as they move, instead of maybe being able to trace one trajectory at a time. Cool. Yeah. So just the summary, right? So we have this regularized continuous normalizing flow that's also the same as optimal transport, and optimal transport is useful because it is a distance between distributions that sort of takes into account, very close distributions. It takes into the horizontal distance between them, not the vertical distance. And so, right, using this, we can define additional objectives that give you, right, optimal transport on the manifold, and you can have cells grow and die, and you can do this all in the neural network space where you can just add additional organizations, and it's easier to parameterize what's happening. And then finally, right, given an individual